Okay, so hello everyone. Today I will present you part of the results I obtained throughout my PhD project, which regards a totally non-controversial topic such as GPCR oligomers. Well, yeah. Okay, so I guess you don't need an introduction about GPCRs. I will just stick to saying that GPCRs are a very relevant target in the pharmaceutical industry. However, some problems are starting to come out in the field of GPCR drug discovery, and the limitations is mostly due to the fact that it's extremely difficult to select, to design selective drugs targeting specific members of this protein family. And so new research avenues are opening up, and most of them are trying to exploit some allosteric regulations mechanisms. And one possible mechanism is the mechanism of lateral allostery. And what this means is that basically you can try to consider GPCRs as oligomers, no? And here is a little bit where the controversy arises, no? Because for class C and class D GPCRs, the dimeric nature is very well established. For class A GPCRs, there's a big debate going on, as you have already seen. Let's say there is some evidence showing that these proteins are able to form transient dynamic interactions, but their functional relevance is not entirely clear for now. And also, since the interactions are very transient, it's also difficult to retrieve a structure of these dimers. However, 4 months ago, a structure of the apelin receptor in the dimeric state was published on Nature Communications. So this is a little bit where we are now. And let's say the aim of my project was to see whether or not the dimerization of class A GPCRs would alter the binding properties of a drug. No? And as an initial use case, we selected the dopamine D2 receptor bound to the well-known antagonist risperidone. And the point was to try and see whether or not this lateral allosteric interaction could alter for instance, the binding affinity of risperidone, or the residence time, or also the binding modes and the intermediate states forming during the process. So initially, we wanted to get some fast insights on this system. So we decided to use some coarse-grained simulation using the Martini force field, and the aim of this protocol was more or less in the same spirit of what we have already seen in the last few days, also what Barbina showed yesterday, where basically you embed your system in a membrane, and then you let the system evolve for very long time scales. Now, since you are using coarse grain, you can go up to hundreds of microseconds or also milliseconds, and you try to characterize your system. And since you have multiple ligands, also the binding is much more probable, let's say. And of course, the bottleneck of coarse grained is always to retrieve the parameters for the ligands, since the force field and the parameters are available for the proteins, but when you want to simulate a coarse-grained ligand, you have to manually retrieve the parameters. Now, first you need to know your molecule, you need to split it into bits, and then you need to make sure that all the bonds, the dihedrals, and the angles are matching, and also the shape of the ligand, of the coarse-grained ligand, should match the shape of the atomistic representation. However, once we obtained once we obtained the parameters for the ligands, we started with our simulations, and we got mainly two insights. The first one was that the importance of the protonation state of the ligand. So first of all, we simulated the system with the neutral species of the drug, and we saw no binding events. No, so here you have a density hotspot map, and the red areas are showing where the ligand is mostly aggregating. And as you can see, there's no aggregation forming in the middle of the helices because there was no binding. But then once we switch into the protonated state, we see density hotspots forming in the middle, and actually we were able to characterize more than 40 binding events. And so we also have a very good characterization of the binding modes, which basically consists in the ligand penetrating the membrane and then infiltrating in between these two helices here, TM1 and TM7. And as a subsequent step, the second insight was that we repeated the same exact protocol for the dimer. And this time what we saw is that the ligand was only binding at one of the two subunits. And so this allosteric interaction between the two receptors was apparently hampering binding at one of the two subunits. As a next step, we wanted to get a more accurate description of the system. So we decided to switch into the atomistic simulations. Now we embedded both the monomer and the dimer into a membrane, and we performed MD simulations. And let's say the aim of this analysis initially was to characterize the binding sites and how the binding site changes when you transition from monomer into dimer. And of course, there were many differences. Most of them were, let's say, very volatile. No, they depend on the frame you select. However, something that stays always consistent is a very big increase in volume. Now that seems to increase when you switch from monomer to dimer, and it elongates in the upper, but also in the lower part of the orthosteric binding site. So, given the differences we found in the binding site, as a next step, we wanted to see whether or not these differences also have an impact on the thermodynamic properties of the ligands. And so we ran some metadynamics calculations, and here the idea was to characterize the free energy landscape of risperidone binding to the monomeric T2 receptor. And then we wanted to repeat the exact same experiment, but this time we added another receptor here as a perturbation, and we wanted to see the differences, right? And so here's the free energy landscape for risperidone binding to the monomeric T2 receptor. What we see is that basically there are two bound states. One of them is perfectly matching the crystal, and the other one is also quite similar, but the ligand is slightly tilted towards the so-called extended binding pocket. At this point, of course, we also wanted to verify the structural stability of the minima, so we performed additional simulations to make sure that the ligand was stable in this pose. And in terms of interaction, they are both quite similar, and they're mostly kept in place by this via this interaction with the aspartate 114. Then at this point, we switch into the dimeric receptor, and we performed the same experiment. Also in this case, we saw two minimum, and also in this case, one of the two was very similar to the crystallographic pose. However, what changes now is that the other minima is completely different, and the ligand penetrates way deeper in the binding site, and this is because now this volume here is accessible in the dimer. Also in this case, we verified the structural stability of the poses, and in terms of interaction, what we saw is that the interactions now change. One of them is still kept in place by this interaction with an aspartate 114, but this other is with aspartate 18. So basically what we show is that we have these two bound states for risperidone within the D2 receptor, and while minimum B is very similar in between the monomer and the dimer, when you switch into minimum A, the pose for the dimer seems to change a lot because the volume is different, and so the ligand is able to penetrate way more inside the receptor. And apparently, this also causes a change in the affinity for the dimer, which results to be higher according to our estimate. And this is a behavior that's already been reported also in the literature. Now, the impact that homodimerization can have on ligand binding. And finally, as a last analysis, we also wanted to get an estimate of the residence time. And so here we used infrequent metadynamics. We performed infrequent metadynamics for risperidone binding to the monomeric state, and then for risperidone binding to one of the two subunits of the dimer, and then the other subunit. And in this case, what we saw is that, of course, that there's a variation when you switch from monomer to dimer, but also there's a very big variation in between the two subunits of the dimer. So yeah, I will go to the conclusion. What I showed is that apparently, according to our coarse-grained simulation, the dimerization of a GPCR can hamper the binding of risperidone to one of the two subunits. Now, when the D2 receptor transitions from monomer to dimer, there's a very big change in volume. This change in volume affects the binding state of the ligand, which in turn affects the thermodynamic properties of the drug. So with this, I would like to thank my group, and yeah, I would like to thank you for your attention.
First, really nice question. Did you try
yeah, that's something we will try now. As I mentioned, there's this new structure of the apelin receptor, but that's a peptide ligand. This is something we would like to do, but that's very, let's say, a very expensive experiments in terms of computation. And so yeah, we are going in that direction also. Yeah, but it will take some time. Yeah.
Yeah. Thanks for the stuff. Have you looked at the one question G protein? And second question, there's some other receptors that have been both dimer and trimer form. I think there's chemokine, CXCR2 and 4.
Yeah. Another direction, yeah, we're taking now is this the impact of the G protein on the dimerization, also because in this apelin receptor structure, they do have one G protein, and they say that it can vary the pose of the ligand binding inside the receptor. So this is something we are looking at. And for the chemokine receptor, I guess there are some papers by the Limongelli group that they are already already worked with this structure, but in terms of computation experiments.
Yeah, because I think with chemokine there's actually a difference in the dimer and the trimeric form. There's some functional studies that show that.
Okay. Yeah, no, I think, yeah, it's mostly a computational experiment, but you mean in terms of measuring the difference in the ligand binding when you go from dimer to trimer. Yeah, I guess it will be difficult there to get the trimeric structure. Maybe you can get some alpha fold. There are structure of the trimeric. Oh, okay, then it should be easier. Yeah.
Once again.